All right. Let's see, I graded the outlines. I think everyone had a good amount of detail. I don't think I wrote on any ones that you needed more detail. So just keep doing what you're doing. That was good. And oops. A reminder, we do have lab this week. So if you're in, I assume Mr. Eagle has quizzes as well, but if you're in my lab on Tuesday or Wednesday, don't forget to take the quiz if you haven't already before lab. So the quiz that was on the material from 2 weeks ago. Okay. So I think we went over this slide, right? I mentioned the organic macromolecules. So we're going to go into more detail on these. Again, these are covered in 1101, Biology 1101, if you took it here, or if you took a general biology course elsewhere, you've probably at least heard these terms. So I'm going to keep it kind of basic. We're not going to go into as much detail as we do in general biology with these. Just kind of a reminder of this is what our building blocks are, essentially. Like when we talk about nutrition in future classes or the different pieces that make up these different organ systems, this is what we're talking about, these macromolecules. So some basics about macromolecules. You're going to want to know the monomers and the polymers for each of these. So a macromolecule is just macro means big. So micro is small, macro is big. So bigger molecules. These are the 4 categories of macromolecules I'll be talking about. We can break each of them down for the most part, into monomers, or we can build them up into polymers. Mono means one, poly means many. So one piece or many pieces. So the monomers are the individual subunits. When you string a bunch of those together, you get the polymer. The polymer is basically those 4 words I showed you before: carbohydrate, lipid, nucleic acid, protein. But I'll talk about what each of the macromolecules are made of on an individual basis, so on a smaller piece by piece basis. Polymers are just repeating units of those monomers. So you, like I said, string a bunch of them together and you get a polymer. A good example, and I'll get into this, is proteins. A protein molecule is just a long string of a bunch of amino acids put together. Have you guys heard of functional groups? Okay, so there's a few functional groups that I want you to be familiar with. In particular for this class, they'll just come up in some of these molecules, and from now on, I'll kind of assume you know what they are. They should look somewhat familiar. Again, they're covered in general biology. A hydroxyl group is really common, and a functional group is basically just a combination of atoms that you see over and over again. So they give a lot of molecules their functions. That's why they're called functional groups. And generally you see these combinations of different elements together. So a hydroxyl group is an oxygen plus a hydrogen, OH. A carboxyl group, you don't really need to know the importance. It's here in case you're curious about it, but I want you to be able to recognize the structural formula if you see it sticking off the end of a macromolecule, which I'll show you in a second. I want you to know if it's a hydroxyl, a carboxyl, etc. So the hydroxyl group is OH. Carboxyl has an OH on it as well, but also a carbon and another oxygen. So a carbon, 2 oxygens, and a hydrogen. But hydroxyl and carboxyl both have these OHs. That's a kind of easy way to remember them. Amino is the only one you need to know with a nitrogen. So N plus 2 hydrogens is amino. Those are going to come into play obviously in proteins with the amino acids. Methyl is a carbon plus 3 hydrogens, CH3. And we'll see that in lipids as well as amino acids. And then phosphate is the only one with phosphorus. So you have your P and then 4 oxygens and it's negatively charged. You don't really have to worry about that. But again, if you see a phosphorus with 4 oxygens, you know it's a phosphate group. Those will come into play in nucleotides, which make up nucleic acids. So those are the 5 that you need to know for this class. I won't ask you any others, just those 5. They won't all come up on the next few slides, or maybe they will. Actually, I think they might. So we'll see them on different molecules and kind of what they look like, and I'll point them out and say, be able to recognize this. If I have it circled, say, on the test, you can tell me if it's a carboxyl or a hydroxyl, etc. Okay, those are the functional groups. Moving on into our first category of macromolecules, carbohydrates. Carbohydrates are pretty simple. They have C's, H's, and O's. So they only have carbon, hydrogen, and oxygen. They're not going to have any other element. And they generally are in this formula. So 1 carbon for every 2 hydrogens, and then 1 oxygen. So for every 1 carbon and 1 oxygen, you have 2 hydrogens. The most common one that you're going to want to recognize is glucose, C6H12O6. So double the amount of hydrogens compared to the carbons and oxygens. They're hydrophilic, which means I think we got to that point on the video, water loving. Yeah. So philic means loving, hydro means water. We'll talk about hydrophobic in a little bit, but these are hydrophilic. They mix with water. They don't repel water. The monomers are called monosaccharides. The polymers are called polysaccharides. Okay, 3 important monosaccharides you need to know. So 3 important monomers. Remember, these will get strung together to make a bigger carbohydrate. Glucose, galactose, fructose. They are all C6H12O6. So if you break down the number of the atoms of each element, it comes out to the same formula. They're just arranged slightly differently. You don't need to be able to recognize them. Like these are the different arrangements of each of them. You don't have to be able to pick them out in a ring form. Just know their names, know that they're monosaccharides, and know that they're all C6H12O6. Glucose, galactose, fructose. Pretty much all of these are going to have oses on the end. I'm going to talk about some disaccharides on the next slide, where there's 2 monomers put together. They're also ose. Yeah, ose usually refers to like sweet. It's a suffix that means sweet. All right, 3 important disaccharides. We've got our table sugar, which is sucrose. You make table sugar by combining glucose and fructose. So 1 glucose molecule plus 1 fructose molecule, you'll get the disaccharide, which is sucrose. Lactose is obviously important. It's the sugar that's found in milk. It's glucose plus galactose. And then maltose is simple. It's 2 glucoses put together, and it's what's used in grain products. So it's used in making beer, among other things. But maltose, all of them have glucose in them, right? So all of them have at least 1 glucose. So it's an easy start. If you know if you're trying to name a disaccharide, it's going to have glucose in it. Then you just have to figure out what the other one is. So sucrose, you have fructose, lactose, you have galactose, which is pretty easy. And then maltose is just 2 glucoses put together. So again, you don't have to recognize the structure. I just have them here for your reference. But you do want to know the two monomers that make up each of these disaccharides. Questions so far? On to the polysaccharides that are important from a human point of view. There's an additional polysaccharide that we went over in general biology, but you don't need to know it because it doesn't occur in us, in humans. So these are long chains of monosaccharides. So we went from monosaccharide, just one little unit of a sugar, to two. And now we're just jumping up to a whole long chain of a polysaccharide. You want to know these three polysaccharides. You don't have to know the monomers that make them up. You just want to know basically their function. Glycogen, first off, is how we store glucose. So if you eat a lot of sugar and glucose gets broken down but not used, your body will store it as glycogen. So it'll string a bunch together and store it. And then when it needs some more sugar or glucose, it'll break it down and release the individual glucose molecules. So energy storage is glycogen. Starch, we don't store starch, but we eat starch. It's a really important nutrient. It's the energy storage in plants. A potato is just a bunch of starch. And then cellulose, also in plants, not in us, but we eat it, and it's a really important source of fiber in our diets. So glycogen is the only one that we actually kind of produce in a way or form. Starch and cellulose are two external sources of polysaccharides that we get for our nutrition. Starch gives us energy. Cellulose basically helps clean out our intestinal system. Cellulose is really strong and we can't really break it down. So cows have, do you know how many stomachs cows have? 4, right? Because they break down the cellulose. We can't break down the cellulose. So they basically send the cellulose through 4 different stomachs to be able to break it down. It's a very tough molecule. Okay, those are the three polysaccharides you want to know. Do you have anything else about carbohydrates? That is it. Any questions about carbohydrates before we move on? That's all you need to know for our purposes and here at this point. Okay. Lipids are our next group. These are hydrophobic. Carbohydrates, hydrophilic. Lipids are hydrophobic. They're going to repel water. So think of oils and fats when you think of lipids. There are 5 categories that you'll need to know. 5 categories of lipids found in human bodies. Fatty acids and triglycerides are very closely related. I'll show you what those look like. Phospholipids, does anyone remember where phospholipids are found? Cell membrane, yep. So all of our cells have a border around them. And phospholipids are most of that border, so they're really important. Eicosanoids and steroids are 2 you may not, well, you probably heard steroids, but eicosanoids you may not have heard of, but these are basically signaling molecules. So they send signals throughout the body. And lipids have more calories per gram of unit mass, I guess, than carbohydrates. So if you eat fats, you get a lot more energy than you do with carbohydrates. All right. First, I'm going to go through each of these 5 types pretty quickly. Fatty acids are basically really long chains of carbon with a bunch of hydrogens coming off. And at the end, you have a carboxyl group. So this is one of those functional groups that I said you'll need to know, the COOH. So you have a carbon double bonded to an oxygen and then a hydroxide group as well. That makes it a carboxyl. And then at the other end, you have methyl. That's another functional group you need to know, C plus 3 H's. Kind of hard to pick out because it just looks like the rest of the chain, but carbon with 3 hydrogens coming off of it is a methyl group. So you have a methyl on one end, carboxyl on the other, just a really long chain of carbons and hydrogens strung together. That's a fatty acid. If you string 3 fatty acids off of a glycerol backbone, this should sound really familiar from general biology, you get a triglyceride. Tri meaning 3. You have these 3 fatty acid chains. So that's what we just looked at. You have the methyl group down here. The carboxyl group gets changed a little bit. So it bonds to this glycerol backbone. You definitely want to know glycerol backbone for a triglyceride. These are really good at storing energy. So they have a lot of energy stored in them. Okay, those are the first 2. On to phospholipids. These are what are found in the plasma membrane of cells, like you guys said. Now we don't have 3 fatty acid chains coming off, but we have 2. We have 2 tails. These are 2 fatty acid chains. You still have some glycerol in there, and then a phosphate group, which makes it a phospholipid. So here you can see the phosphate, the P plus 4 oxygens. That's one of the functional groups you need to know. Glycerol, and then 2 chains of fatty acids. We'll talk about phospholipids in the next chapter when we get into more details about the cells, yeah. Okay. Next, we have a couple of groups that you might not be as familiar with. Eicosanoids, you probably haven't heard of before you did your outline. If you put them in your outline, these are, again, long chains, so 20 carbon chains, usually with a ring at one end. So we haven't seen a ring yet, but here you can see basically a carbon ring. So these, if you haven't had organic chemistry, this is how carbon rings are typically depicted. Each of these intersections here where 2 lines come together are a carbon. Same with this. Just because there's so many carbons in organic chemistry, they don't draw out all of the C's. All of those intersection points are carbons. Okay, so you have chains of carbons with a ring at one end. That's how you'll identify an eicosanoid. And like I said, these are important signaling molecules. So they're lipids that send signals around the body. And you can see here some of the examples where they're used, blood clotting. So if you get a cut and your blood needs to clot at that opening, eicosanoids will be sent there to start the process of blood clotting. Hormone action. Hormones we'll talk about on the next slide as well. They're steroids. A lot of them are steroids, I should say. But eicosanoids can signal the release of different hormones in the body. Labor contractions, that goes back to hormones as well. So eicosanoids are important in developing labor contractions and then changing blood vessel diameter. So if you shrink down a blood vessel, that's going to increase the pressure, the blood pressure. If you dilate a blood vessel, that's going to decrease the blood pressure, just like a hose. So eicosanoids are really important in maintaining a stable blood pressure. Questions? Am I going too fast? Keep up. Okay. Finally, we have steroids. They're also referred to as sterols. These have rings with kind of a tiny tail. So multiple rings, usually four carbon rings with a little hydrophobic tail sticking off the back end. Cholesterol is our main steroid. All other steroids are made from cholesterol. So cholesterol will be changed into other steroids. So any other steroids in your body, you have to have cholesterol in the first place to make those steroids. So it's the parent steroid. Important for a lot of things, particularly in the nervous system. When we get into the nervous system, we'll talk about why cholesterol can be really important. It's also found in cell membranes. So you have all those phospholipids in cell membranes. Interspersed in between those, there's cholesterol molecules. So these little guys sticking out of there, which helps solidify the membrane a little bit, makes it less liquidy. Some other examples. So if you start with cholesterol, you can build cortisol. Does anyone know what the common name for that hormone is? Kind of. It has to do with fat. Stress. Yeah, it's the stress hormone. So cortisol, if you're stressed out, your body is creating a lot of cortisol, which can lead to more storing of fat in the body. Progesterone, estrogen, testosterone, those are all sex hormones. And they all come from cholesterol. All right, we're cruising along. Any questions about those? I'm not going to go into good and bad cholesterol right now. That's more for AMP2. I just don't have time right now. That's all you need to know for cholesterol at this point. On to proteins. So proteins are unique in that now we have an amino group sticking off of them, right? So we haven't had nitrogen in any of the other macromolecules we've talked about. So now we have nitrogen. These are nitrogen-rich structures, and each protein is made up of a bunch of amino acids. So amino acids are the monomer. The protein in general is referred as the polymer. Altogether, there are 20 different amino acids. The order in which they're strung together is going to determine what protein eventually results. If we look at an amino acid, they all have a really similar structure except for one piece. So this R side chain is just kind of a space holder. That's what determines what amino acid it is. Otherwise, all of the amino acids have, well, a central carbon, a carboxyl group. So there's our COOH again. That should look familiar. A hydrogen up here on its own, and then an amino group. So that's our NH2. Another functional group you should be familiar with. So you have amino, carboxyl, and then this R side chain is what makes each amino acid unique. All of the 20 amino acids have a different chain of molecules coming off of here. We're not going to talk about what they are, what the different chains are, but just imagine a whole lot of other atoms coming off of here. It's just, like I said, it's labeled R just to kind of save the place. Proteins are obviously very important in our body. They have a lot of functions. I am listing some here. I don't have all of them listed, but these are the ones I want you guys to be familiar with. First off, they're very important when it comes to structural components of our body. Keratin and collagen are two structural proteins that you may have heard of. They're all the rage in like shampoos and skincare products these days. So keratin is really important in providing strength for hair and nails and skin surface. So that's really a surface layer protein. A little bit deeper, you have collagen, also a structural protein. It's in skin, bones, cartilage, teeth. So when we talk about, well, this week in lab, we're going to get into some tissues. You'll see collagen fibers in the cartilage tissues that we look at. Similar to some of the lipids I mentioned, proteins can send messages around the body. So they'll send information. Some hormones are proteins, and hormones are just chemical signals. And there's other kind of cell-to-cell signaling molecules that I won't get into. On the surface of cells, those messages have to be taken in somewhere. So those are usually proteins on the surfaces of cells that will bind to hormones. So hormones will bind to the surface protein, and that's how the message gets into the cell to tell it to do whatever it needs to do. They help transport things across the membrane. So they'll bind to hormones on the outside. They'll also transfer stuff into or out of the cell. We'll talk about that in the next chapter in a little bit more detail. But they determine what goes in and out. They're like the gatekeepers of a cell. And then they're really important as enzymes or catalysts. So they will speed up the rate of a chemical reaction. I'm not going to go into too much detail right now about how that works, but they make chemical reactions more efficient and faster. If enzymes are present, these reactions can happen more efficiently. And enzymes are proteins for our purposes in our bodies. Any enzyme is going to be a protein. And they're important in recognition and protection. So there's proteins on the outsides of cells that will recognize invading viruses or bacteria and elicit an immune response. So they're really important in our immune system. Antibodies are proteins. Antibodies are flowing through our blood, trying to find any invading organism or virus that might be invading. And particularly pertinent these days, they've identified a protein on the outside of the SARS-CoV-2 viral cell. So viruses aren't technically cells, but we'll call it a cell. So it's a really complicated protein. This is what allows the SARS-CoV-2 virus to get into an individual's cells. So without this protein on the outside, it wouldn't be able to invade our cells and make us sick. So identifying that was a huge step forward. They've known about it for a little while now, but just an interesting kind of really recent application of this external cellular protein idea. And that's how a lot of times the vaccines will actually recognize this spike protein on the outside of the viral cells. And so that's how our bodies can recognize it and then destroy those viral cells. All right, that's all I have for proteins. On to nucleic acids. Nucleic acids are made up of nucleotides. So a bunch of nucleotides strung together makes a nucleic acid. The only two nucleic acids we're talking about or that exist are DNA and RNA. Nucleotides are sort of complicated monomers. They have three different parts. So they have a nitrogenous base. That basically means there's a lot of nitrogen in it. And I'll show you what this looks like on the next slide and point out on a diagram where these are in a nucleotide. There's a monosaccharide. What is that? You guys remember we just went over the category of macromolecule that a monosaccharide is. That's right, it's a carbohydrate. So it's like glucose, for example, a monomer of a carbohydrate. And then you have a phosphate group or maybe more than one phosphate group. And we know phosphate, that's PO4. That's what makes up one nucleotide. Then you string a bunch of those together and you get RNA. If you bring two of those strings together, you get DNA. So it's a pretty complicated polymer. ATP, I think most people have heard of, yes. The energy source for our cells, that is a nucleotide. ATP stands for adenosine triphosphate. So how many phosphate groups do you think it has? 3, yeah. So 3 phosphate groups, and I'll show you, I think I have an image of what it looks like maybe. Anyway, ATP is the most famous nucleotide. Most other nucleotides, we don't really talk about individually. This is like one monomer of a nucleic acid, just on its own. It doesn't string together to make DNA or RNA, it's on its own. So I want you to be familiar with that. Know it's the main energy source for cells, know what it stands for, adenosine triphosphate, and know that it is a nucleotide. We won't talk about any other individual nucleotides, just ATP. Okay, so let's look at an image of these. Here's ATP, perfect. So ATP is like I said, a nucleotide. We have 3 phosphate groups. The nitrogenous base is up here. You can see the nitrogens in there. You don't need to know the name of it. But nitrogens in a couple of rings, it'll either be 1 or 2 rings in a nucleotide. And then ribose, ose, should make you think sugar, carbohydrate. So this is our monosaccharide, ribose. There's another nucleotide over here. If you want to look at it, it only has 1 phosphate group, but it still has the nitrogenous base and the sugar. Okay. So what does, I don't think I have DNA and RNA on here. What does DNA stand for? Anyone?
Deoxyribosome something.
Close. Deoxyribonucleic acid. Just have to pull it from everyone. Deoxyribonucleic acid. So deoxy we won't worry about, but ribo refers to ribose, that's the sugar. And then nucleic acid, obviously it's a nucleic acid. Awesome. And then RNA is just ribonucleic acid. Not ribosomal, but ribonucleic. Yep. Ribonucleic acid. Okay. Those are the only 2 that you need to be familiar with. Not really going to ask you a whole lot about nucleic acids. And now, okay, so put your notes away. This is not for a grade, but I have a little mini quiz to see how much you guys picked up from that or how much you remember. Not for a grade, put your notes away. I think it's like 5 questions, 5 or 6 questions about the organic macromolecules. Let's see how many we can get. All right, so first off, you can write them down or not. I don't have this slide on Canvas, but I'll put it up there after class today. All right, so triglycerides. Well, what are they first off? What kind of major macromolecule? A lipid. So they're lipids. There are 3 fatty acids attached to what? A glycerol backbone. So you definitely want to know this. 3 fatty acids attached to a glycerol backbone are triglycerides. What is the monomer of a protein? Amino acid, yeah. Amino acid. These are the kinds of questions I would ask on, say, an exam, but it would be multiple choice. But still, this will give you an idea as to how much you're picking up. Okay, which type of carbohydrate category does lactose belong to? Mono, di, or polysaccharide? Disaccharide, yeah. Who remembers what 2 pieces make it up? 2 monomers. And galactose, yeah, that's pretty easy. So it's always glucose in any of the disaccharides. And then it's lactose. So the other one is galactose. Cool. And it is a disaccharide. Which of the four main categories of macromolecules contain phosphate groups? What, lipids? Lipids, wait, phosphate now, not lipids. Lipids? Not lipids. What did we just talk about? Yeah, nucleic acid contains one or more phosphate groups. And then what else? Lipids? Anyone? I'm blanking. I want to say proteins. Let's go see. Why am I blanking on this? Let's go back. So we know not proteins. Phosphates, yeah, phospholipids or lipids. Nice. Sorry, I totally blanked on that. See? Okay, lipids, phospholipids, and nucleic acids. My bad. Okay, and then enzymes. What are enzymes? Yes, they are proteins. Okay, sorry to throw you off about the phosphate groups. Okay, I will put this slide. This slide has the answers at the bottom in the little note section. So you can go back and kind of relook at these if you didn't get those answers down. Okay, that wraps up the chemistry lecture. Any questions about that? Okay. So yeah, the answers are down here. All right. We're going to go into cell structure next. So I'm just going to barely get started because we have some time. I want to make sure we have enough time to review before the first exam. So in the interest of that, we're going to keep going. All right, cell structure. All right, into chapter 3, cell structure. So you haven't had to turn in your chapter 3 outline yet, so some of this might sound new, but we only have like 10 minutes, so we won't get that far into it. And hopefully some of it sounds kind of familiar. So first off, cell theory. So the idea that these 3 components are really important in the development of life, essentially, is what the cell theory is kind of getting at. So, oops, forward. First off, the 3 tenets of the cell theory, all the 3 that we're going to focus on. All organisms are composed of cells. So all of us are made of at least one cell. We're many, many cells, obviously. So the cell, by definition, is the simplest form of life. So a bacteria is one cell, but it's still considered alive. So you have to be made of at least one cell for us to consider something alive. And then cells make more cells. So you can't have a cell develop out of nowhere. There has to be a cell there to basically split and create another cell. Those are the 3 principles that you want to be familiar with for the cell theory. We have many, many types of cells in our body. And this is what we're going to be getting into in the next big chapter and in lab this week with tissues. There are all kinds of different cells, different shapes, different amounts of layers, different functions throughout our body. So we have almost 200 types of cells in our body. The cells that make up our skin are very different from the cells that make up our blood, for instance. Yeah. So we'll talk about those and how those cells work together to form tissues. That's kind of where we're going with this, but we have to know how a cell sort of works before we get into how tissues work. The cellular components. So these are the main pieces. There's others, but I'm not going to have you really be all that familiar with them. We'll go into some of the organelles towards the end. But these are kind of the main pieces if you think of a cell, kind of stepping back. Plasma membrane, we're going to talk about a good bit. That's the border around the cell. And like I said, made of proteins and lipids, primarily. The cytoplasm is kind of everything within the cell. Cytoplasm and cytosol are sometimes used interchangeably. Cytosol is technically all the fluid that's in the cell. The cytoplasm includes all of the organelles. So all these little pieces in the cell would be in the cytoplasm, part of the cytoplasm. When we talk about tissues, I'll be mentioning ICF and ECF. ICF refers to intracellular fluid, so the fluid inside the cell. Extracellular fluid is obviously everything outside the cell. So we have fluid in our cells, we have fluid outside our cells. A lot of the movements of things will depend on the gradients between what's in the cell in the ICF and outside the cell in the ECF. Interstitial fluid is another term for extracellular fluid. Interstitial means in between the tissues. So when we talk about tissues, there's fluid in between the cells that make up the tissues as well. So you'll see interstitial. Okay. So ICF and ECF, that'll come back up. First, getting into the plasma membrane. I think this is the exact same slide I use in general biology, so hopefully it looks kind of familiar. Most of the molecules that make up the cellular membrane are lipids. 98%. The other 2% are essentially proteins. So most of the molecules by number, if you're counting them, are phospholipids. 75% of the 98% is essentially phospholipids. There's some other lipids in there, like I mentioned, cholesterol. So by and large, we're talking about phospholipids making up most of the membrane of a cell. They're really important in a couple different ways. So you have a bilayer of phospholipids. So you have, this is usually how phospholipids are depicted. A head, which is hydrophilic, and then the tails are hydrophobic. Oh, there you go. That makes them amphipathic. Amphi meaning both ways, like an amphibian lives in the water and out of the water. These are amphipathic, meaning there's a hydrophilic side and a hydrophobic side. That's what allows them to, that's what makes them a nice border for the cell. And then being in a bilayer means that the hydrophilic heads are always facing away from each other. So if this is a cell membrane, we'll imagine this is inside the cell, intracellular. This will be outside the cell up here. The heads are facing away from each other. The tails are facing towards each other. And the tails face inward because they want to be away from the water. They're hydrophobic, right? So this is a nice little hydrophobic layer in here. So water can't just pass through willy-nilly. Anything that's hydrophilic can't pass through here very easily. It needs some help. We'll talk about how that works in a little bit. So it's a good gate to keep most molecules out and only allow the ones in that they want in, and then vice versa. Okay. Hydrophilic, hydrophobic. So yeah, the hydrophilic head, that's the phosphate group. Hydrophobic tail is going to be the fatty acid chains. We already talked about that. So you get this bilayer with a hydrophobic interior portion. The other lipids, you have cholesterol and glycolipids. So cholesterol, I mentioned, it makes up most of the rest of the membrane lipids. So phospholipids are like 75%, cholesterol is about 20%. The important part of cholesterol in our cell membranes is that they hold the phospholipids in place. So without these little cholesterols interspersed, which you see here, the little blue 4-ring structures. Those are cholesterols kind of interspersed in there. They just hold the phospholipids in place. Otherwise, the phospholipids would be flipping over each other a lot more frequently and be a lot more liquid. So they help stabilize the membrane. Too much cholesterol is obviously a bad thing. You don't want a really stiff cell membrane. You want kind of somewhere in the middle, 20%. That's the ideal. And then glycolipids, you may not have heard of before, but they are lipids with a carbohydrate chain attached. So glyco refers to anything dealing with like carbohydrate. You can think glucose, glycogen, etc. So short little chains of carbohydrates attached to a lipid, attached to the phospholipid specifically. And they're kind of like flags or little signaling molecules to help one cell communicate with another cell. So they stick out, they can take signals in or send signals out, or help cells realize what else is next to them, etc. So they're important in cell-to-cell communication. And then proteins. We're not going to get into proteins today. I'm going to stop here. But like I said, they make up about 2% of the number of molecules, but they're really heavy. So 50% of the mass. We'll get into them in more detail on Wednesday. So we'll stop there, and I'll see y'all in lab Tuesday or Wednesday. Any questions?